but still no. Yeah, finally. Okay, let's begin this session. So the next speaker is Yunbo Shi. Yunbo did his bachelor's in Wuhan in China. Then he moved to UC Berkeley for his PhD. Subsequently, he eventually moved to NICHD in, if I'm right, in 1992. And ever since, Yunbo is there at NICHD. He's currently the Deputy Director of National Institute of Child Health and Disease at NIH. And he has been working on thyroid hormone metamorphosis using Xenopus as a model for a very, very long time. Thank you, Yunbo, for agreeing to give a talk here. We are looking forward to it.
Well, thanks for the kind introduction. I appreciate this opportunity to participate in this very interesting meeting. I have enjoyed and learned a lot, and I would like to thank Project and other organizers for inviting me to speak here. As my title suggests, I actually will be talking about frog metamorphosis, which deviates from regeneration, the main theme of the meeting. But frog metamorphosis is actually highly relevant to regeneration, because many tadpole tissues and organs actually lose their regenerative capability during metamorphosis. Thus, understanding metamorphosis should shed light on how regeneration is controlled in development and perhaps in evolution. So to begin with, as an introduction, I would like to divide human life cycle artificially into 3 phases by using the levels of our favorite molecule, thyroid hormone. As you can see here, during the first 5 months of human embryogenesis, there's very little thyroid hormone present in the plasma. And this is the period when many organs and tissues are initially formed. The second period is a so-called post-embryonic development. This is when thyroid hormone level rises, in the case of human, peaks around birth, and then drops to a steady state level several months later. This is the period when many organs and tissues mature into their adult form. And finally, what we call child growth and adulthood. This is when thyroid hormone level is a steady state. Now, of the 3 phases, post-embryonic development is the least studied, largely because embryos and the neonates are dependent on maternal supply of nutrients for survival, making it very hard to manipulate. On the other hand, defects during this period actually contributes to many diseases that persist throughout adult life. For example, thyroid hormone deficiency during this period will lead to severe developmental defect. For example, shown here is one example. A person is much shorter than an age-matched person. In addition, there's also severe neurological disorder. So how does thyroid hormone actually control this developmental period? Now, we thought actually frog metamorphosis, shown here is a Xenopus metamorphosis, may be a good model for this. As you can see here, during the 2nd month, that's roughly day 30 to day 60, or between stage 54, 55 to stage 66 in Xenopus, the tadpole is converted into a froglet. And this period has very high levels of thyroid hormone in the plasma, very similar to what happens during post-embryonic development in human or in mammals in general. Importantly, many organ transformation processes are very similar between here and here. But metamorphosis can be easily controlled. For example, we can take a tadpole as soon as it's formed, roughly 4 to 7 days old, and then when feeding begins, we can actually add physiological levels of thyroid hormone to the rearing water. And within a few days, you will see metamorphic changes occur. And naturally, this will take several more weeks before metamorphosis is initiated. In addition, if we take any tadpoles before metamorphosis begins, and then we block the synthesis of endogenous thyroid hormone, the tadpole will never undergo metamorphosis. It will stay as a tadpole for several years at least. Although natural development occurs within about 2 or 3 months to become a froglet. So this indicates thyroid hormone is both necessary and sufficient. And more importantly, without thyroid hormone, the tadpole will eventually die before reaching productive age. That is, remain as a tadpole and then eventually die, even though live much longer than a normal tadpole. And this actually is very similar to what happens in mouse. Genetic studies suggest that if you block thyroid hormone synthesis entirely in the developing mouse, the mouse will die shortly after birth. So this indicates both in frogs and in mice, thyroid hormone is essential for the animal to develop into reproductive age or sexual maturity. Now, it is very likely the same thing is true for human, because so far there have been no reported human cases where there's no thyroid hormone at all. So then the question is, how does thyroid hormone control this developmental period? Now, as you have heard from Prayag's talk earlier, he gave very nice introduction on the thyroid hormone. We know thyroid hormone is synthesized by thyroid gland, is secreted into the circulating blood. Then the hormone is carried over to different tissues where it migrates into the nucleus. In the nucleus, it binds to the so-called thyroid hormone receptors, or TRs. TRs are nuclear localized transcription factors. And TRs actually can form dimers with ARXR, or 9-cis-retinol acid receptor. ARXR, TR are the same family of transcription factors that bind to hormones. So we know that in eukaryotic cells, the genetic material, the DNA, is complex with proteins called histones. DNA histone complex form a unit called a nucleosome. Such nucleosomes then form a complex structure called a chromatin, and in turn lead to the formation of so-called individual chromosomes. What we and others have shown is that TR-ARXR heterodimer, shown here, can bind to specific DNA called the TRE, or thyroid hormone response element, localized in the chromatin. And this binding then brings other proteins, so-called co-repressor complexes, to this region. And often such co-repressor complexes containing enzymes that can modify the histones, leading to the formation of the so-called repression marks, that is, histone modification associated with gene repression or gene silencing. At the same time, the so-called activation histone marks, the pink dots, are reduced by such enzyme complexes. So we believe such modification then help the unligated receptor complex to keep the gene repressed. So therefore, no mRNA is made from this promoter region. Now, when thyroid hormone is present, the hormone then binds to the receptor, which in turn brings different set of proteins to the binding site. These are so-called co-activators. This in turn leads to opposite change, as in the case of no thyroid hormone, that is, increased level of activation histone marks and reduce the repression histone marks. In addition, we have found that up to 2 to 3 nucleosomes were disrupted. That is, the histones in this region are removed from the DNA. So we believe this kind of chromatin remodeling and histone modification plays a critical role in how thyroid hormone receptor regulate transcription. So in this case, the transcription from this promoter is strongly activated. So this model was initially worked out from in vitro and cell culture studies. So a key question we have is, do thyroid hormone receptors play 2 different roles in development? That is, unligated to repress gene and ligated to active gene during development. So a number of years ago, actually we proposed this dual function model for thyroid hormone receptor during frog development. That is, we hypothesized that thyroid hormone inducible genes, shown here, are expressed at a basal level during embryogenesis, that is stage 1 to 45, roughly 4 to 7 days old in Xenopus. And this is the period is called basal because there's no thyroid hormone receptor or very little, and there's very little thyroid hormone present. So we believe this expression is maybe important for embryonic organ development. By stage 45, a tadpole is formed, animal begin to feed. So development is actually complete. So therefore, we can imagine those genes are no longer needed. They should be silenced. So we propose the expression of TR alpha gene and ARXR alpha gene will form heterodimers that bind to the promoter region in chromatin, and it keeps genes repressed because there's no thyroid hormone or very little thyroid hormone during this pre-metamorphic period. And we believe this repression then is to make sure the animal doesn't undergo metamorphosis too early. So we want to make sure to grow for several weeks. Okay. Now I should have pointed out, in all vertebrates, there are actually 2 TR genes, TR alpha and TR beta. The TR beta gene has very little expression early on, but is activated during metamorphosis when thyroid hormone is available. So actually, the receptor activated is only expression. TR beta gene's expression is activated. There are actually 3 ARXR genes, but ARXR alpha is the one highly expressed throughout development. Then after stage 54, which is considered the onset of metamorphosis, endogenous thyroid hormone synthesis occurs. This then leads to the formation of a liganded receptor to activate transcription. And we believe this activation is important to initiate metamorphosis and make sure the metamorphosis progress properly to the end of development, stage 6 to 6. So over the years, we and others have carried out different molecular genetic studies to test this model. And what I would do today is to summarize some of our recent studies by using gene editing technology where we can knock out the endogenous TR alpha and TR beta gene. This is sort of complement the overexpression studies used by using transgenesis, as Prayag talked about in his talk. So what we first did is actually focus on TR alpha, because TR alpha is highly expressed during pre-metamorphosis and throughout metamorphosis. So we think TR alpha may have a more important role in regulating pre-metamorphic development. So our prediction is that if we take out TR alpha during pre-metamorphosis, then the genes won't be repressed, then become expressed similar to embryogenesis. And this then may make the animal to enter metamorphosis or initiate metamorphosis prematurely. So is that the case? It's actually very easy for us to do for frog development. All we need to do is knock out the gene, okay, knock out this alpha gene, and then measure how long does it take to go from fertilization to stage 54, which is the onset of metamorphosis. We can also measure how long does it take to go from stage 1 to stage 45, embryogenesis. Suffice it to say, TR knockout doesn't do anything to embryogenesis, as far as we can tell. What happens to pre-metamorphosis? What we found is that wild-type animals takes about 45 days to go from fertilization to the onset of metamorphosis. However, TR knockout homozygous. Homozygous means both copies of the genes are removed. One comes from male, one comes from the female. They're both removed from the genome, whereas heterozygous means one of the copies still wild type. So this indicates TR knockout indeed alters the timing of metamorphosis. Interestingly, during our study, we noticed another unexpected phenomenon, that is growth rate. If we take all animals and then measure the size at a given day, that is all animals are the same age, and then we artificially divide them into 3 groups, small, medium, and large, based on the length of the tadpole. What we found is that wild type mostly are medium in size, However, homozygous knockout are mostly large. So this indicates removing TR-alpha makes the animal grow faster in size. And furthermore, we found molecularly, this appears to be regulated due to increased growth hormone gene expression. So its homozygous knockout has higher levels than the wild type for both genes. There are two genes for growth hormone genes in Xenopus, but there's only one in mammals. Interestingly, in human or in mouse, the growth hormone gene is a thyroid hormone target gene. This would be consistent with what we observe here. That is, in the pre-metamorphic tadpoles, growth hormone gene is bound by receptor, which is unligated, consequently is repressed. When we get rid of the growth hormone, the thyroid hormone receptor, the gene is de-repressed, leading to faster growth. So this indicates in pre-metamorphic tadpoles, thyroid hormone receptor alpha has two functions, regulating growth rate and developmental rate. So are the two related? The reason we ask this question is because, as many of you may know, in some animal species, such as Drosophila, development from some phases to the next requires the animal to reach a minimum size. If you don't reach that size, they will just stay there, will not change. So we wonder whether that's the case in frog. If so, we can imagine the faster growing tadpoles will reach that minimum size sooner, then it can undergo metamorphosis. Right? If that's the case, we would predict animals at the onset of metamorphosis should be similar in size. Is that the case? Well, it turns out that's not. If we allow the wild type and knockout animals to develop, regardless of how long it takes, we allow them to develop to stage 54, that's onset of metamorphosis. Then we measure the size, the length. What we found is that wild type animals are actually twice as long as the homozygous knockout. So this indicates unligated thyroid hormone receptor have two functions, regulating growth rate and developmental timing. But these two functions are not dependent on each other. They may be independent of each other. So then we ask what happens later on. That is, what happens when thyroid hormone is available? We initially focus on the early metamorphosis between stage 54 to 58. That is when thyroid hormone is first detectable, to 58, which is considered the beginning of metamorphic climax. We focus on this, the ending at this stage, because by now, TR beta level is getting quite high, which may be able to compensate for TR alpha. So we want to find out what's the role of TR alpha during this period. So again, we just measure the time it takes to go from here to here. What we found is that unlike pre-metamorphosis, now the knockout slow down development. It takes twice as long, more than twice as long in this case, compared to wild type animals. Heterozygous sits right in between, which makes sense. So then what happens, we ask what happens later on. As I said earlier, by stage 58, TR beta is high. So can TR beta compensate for the loss of TR alpha in the knockout animals? And so we measure the time it takes between 58 and 66. We found knockout doesn't do anything. So suggesting TR beta indeed can compensate. Furthermore, we also measure how long does it take to go from stage 1 to the end of metamorphosis in a wild type and knockout. And we found wild type, homozygous knockout, or animals with one copy of the receptor alpha, that is heterozygous knockout, that by and large all the same. So this actually is kind of a puzzling. Why do you need a receptor? Because you started out without receptor, ended up without receptor, the animal developed fine, reaching the end of metamorphosis. So if you will look carefully in development, what we found is an interesting phenomenon. That is, first, if we look again, stage 45 is the end of Amphibian. Wild type and knockout are similar in size and age, so there's no developmental defect at all. Once tadpole feeding begins, at the same age, that is, if you draw a line down this way, Same age, the knockout animals always grow faster. It's larger in size. But it develops really fast, reaching the onset of metamorphosis, stage 54, at a much younger age compared to wild-type siblings. And the wild-type siblings, even though they grow slower, but because it has extra time to grow, so by the time it starts metamorphosis, it is actually bigger than the knockout sibling. And once metamorphosis begins after 54, the wild type goes very fast to reach the end. No, I'm sorry, the wild type goes very fast to reach the end, whereas the knockout takes a very long time to reach there. So even though they both reach the metamorphosis at the same age, but the metamorphic time is much longer for the knockout. And furthermore, the animal started out in the knockout as a smaller tadpole to start metamorphosis, also ended up smaller frogman. So why you need to do that? It turns out actually perhaps this is very important for development, animal survival, and perhaps evolution. Because we know as the animals evolve from aquatic living to terrestrial living, eventually there's only one phase. You don't have a biphasic development like in amphibians. You form a tadpole first, then become a frog. In mammals, you develop directly into the adult form. So the metamorphic phase is eliminated. But you notice with TRα, the metamorphic phase is shorter. And whereas without alpha is longer. And this longer also is not good for animal survival. We know during metamorphic period, the animals are very vulnerable to two types of danger. One is actually predators. If you're smaller in size, take too long to change, there's more chance being killed or eaten by predators. Environmentally, condition could change, the pond could dry. So if you take too long, it's possible the tadpole hasn't finished metamorphosis before the water is gone. Then they will die. So we believe the thyroid hormone receptor alpha functions to ensure the animal reach the right size and then go through metamorphosis quickly. So that is shorten the time needed for metamorphosis and making them larger so it can survive better. So therefore, even though overall development is not changed, but the survival and the shortening of metamorphic timing, which may be important for evolution, are critical, and they're regulated by TRα. So as I said earlier, even though without receptor alpha, the animal can finish metamorphosis, suggesting it may be compensated by TRβ. So what is the role of TRβ? So we actually knocked out both alpha and beta individually. Now I will show you the difference by using 3 organs for comparison. So stage 54 is the onset of metamorphosis, okay? If we look at limb development, which occurs very early, before metamorphosis, that is when there's no thyroid hormone, TRα knockout accelerates this process. So the animal initiates metamorphosis sooner. Whereas once metamorphosis begins, TRα knockout slows them down, slow this process down. Whereas TRβ knockout doesn't do anything here. Intestinal remodeling, which changes a tadpole intestine like this to a frog intestine like that, and occurs mostly between 58 and 66. And during this period, both TRα knockout, TRβ knockout slow down this process. Finally, tail resorption, which is most noticeably after stage 61, when the tail shrinks. Eventually, no tail left at all. What we found is that TRα knockout doesn't do anything, whereas TRβ knockout slow down this process. And furthermore, we found in general, the effect of TR knockout, individual TR genes, correlate very nicely with the relative expression level of individual TR genes. That is, we don't think TRα, TRβ are intrinsically different. What makes them have different function is because they are expressed differently during development in different organs. And as long as you have one of them present, eventually they can compensate each other to make sure that metamorphosis is complete. So then we ask, what happened if we get rid of both receptors? So I will only summarize very briefly our findings. If we do that, and simply measure how long does it take for the animal to go from stage 1 to 54, that is onset of metamorphosis, we found TR double knockout, that is remove both TR genes, make the animal develop very fast, okay, just like TR alpha knockout alone, okay? And then once metamorphosis begins, again, like TR alpha alone, the tadpole, double knockout tadpoles takes forever, very long, to reach the climax of metamorphosis, stage 6 to 1. Twice as long compared to the wild type animals. And so this indicates receptor indeed controls metamorphic timing before metamorphosis and metamorphic rate after metamorphosis. So then the question is, what happens after stage 6 to 1? What we found is that TR double knockout tadpoles, that is without either receptor, they all died around stage 60 and 6 to 1. You can see here, 100% dead around stage 6 to 1. On the other hand, wild type tadpoles, or tadpoles with even a single copy of TR beta gene, that is no TR alpha, 1 TR beta, or 2 TR beta genes, but no TR alpha, can develop past 61, and then eventually become reproductive adults. So what happens at this stage? So what happens, if we look carefully, is that at stage 61, if you compare the wild type to knockout tadpoles, what you see is that the limbs and the head structure, the body structure, are very much similar between the wild type and knockout tadpoles. In fact, all the structures are adult-like, suggesting removing the receptor, that is de-repression of the genes, is sufficient for adult organ development. Now, if you look at the gills, you see the gills here, the two gills are much larger than the wild type siblings. Gills are larval-specific organs. So that means gill resorption is inhibited by TR knockout. Tail, which is the largest larval-specific organ in a frog or tadpole, what you see is that stage 6 to 1, tail resorption has not begun. Tail length reduction occurs around stage 6 to 2 and after. So we ask, what happens if we allow the animal to develop? What we found is that in wild type animals, within 1 week, okay, within 1 week, the tail is completely gone. The gill finish as well, resorption. However, the knockout tadpoles remain this morphology for average about 2 weeks. That is longer than this 1 week needed here, and then they die. So then this suggests to us that larval tissue degeneration does not occur in the absence of thyroid hormone receptor. So the receptor is more important for larval tissue regeneration than adult organ development. Although it does control the rate of adult organ development, but it's just not required, not essential. So this then leads me to the summary slides on the function of the receptor. That is, we and others have carried out different molecular and genetic studies that really support this dual function model for thyroid hormone receptor in development. And in particular, the most recent gene editing studies not only support the role of the receptor, but also fill in the more details of the receptor function. That is, what we found is that TR alpha has a more important role in regulating the timing of metamorphosis. And in addition, it also has a role to regulate the growth rate. Okay, and I want to add at this point, this suggests that thyroid hormone regulates growth rate and metamorphic timing independently, our evidence suggests. This actually is quite similar to what we observe in mammals, or particularly in human. We know human in different regions sometimes are very different in size at same age, but developmental, like such as puberty, sexual maturity, is controlled more by age than by size of the human. So suggesting that perhaps in mammals, thyroid hormone receptor may also have such function that is regulating both developmental rate and growth rate. That remains to be tested. Now, once metamorphosis begins, what we found is that both TR alpha and TR beta contribute to the rate of progression between stage 54 and 58. However, the most important role of the receptor is really to ensure larval tissue resorption, especially around the climax metamorphosis and then later. So the question is this unique to frog development? Well, I will use my last slide to make a comparison and perhaps to raise some potentially testable hypotheses about the role of thyroid hormone receptor in development in general in vertebrates. That is, as I mentioned earlier, if we compare mouse development and frog development, what we see is that thyroid hormone is critical for post-embryonic development or metamorphosis. Without thyroid hormone, we need lethality of the animals prior to become reproductive adults. Okay, this occurs both in frog and in mouse. We have genetic evidence to show that. But in human, we do not know yet. However, it's most likely similar since no human has been found to be totally lacking thyroid hormone. Secondly, we found individual thyroid hormone receptors are totally dispensable, although individual knockout can alter development, leads to some alterations and defects in both mammals and frogs, but the animal will survive and can reproduce. And finally, the third one is perhaps the major divergence of thyroid hormone receptor function during development, during evolution. That is, we found mouse with both TR genes knocked out can develop into adult. However, Xenopus tadpoles without TR will die at the climax metamorphosis. So, why there's such a divergence? In fact, actually, I found this divergence is perhaps the most interesting with regard to the role of thyroid hormone receptor in evolution. Okay, if you look carefully, as I showed you earlier, TR is not required for adult organ development in tadpoles, okay, but is essential for larval tissue resorption. In addition, Xenopus, but not a mouse, has many larval organs to be resorbed in order to become a frog. That is because in evolution to mammals, this second phase of development, that is a tadpole stage, is eliminated. So a tadpole, the embryo is directly developed into adult structure, adult form. And because of the elimination of this second phase of development, we don't have larval specific organs that have to be resorbed, removed to become an adult. And this perhaps is the reason why in mouse, without TR, the animal can develop into adult, because there's no larval organ to remove. With adult organ, development is conserved. TR is functioning, is not essential, but it can regulate how fast the organs mature into adult structure. So we think maybe perhaps this evolutionary change in TR function may be important during evolution for the elimination of the second phase of the frog development in order to become terrestrial animals like mammals. So with that, I will end my talk, and I would like to point out the work that I presented here today actually involved a number of current and former members in the lab. I won't point out details one by one. I just want to point it out our knockout study was initiated in collaboration with Dr. Yongwen Chen's lab when he was in Guangzhou Institute of Biomedicine and Health in China, particularly his technician at the time, Xiaogang Guo. And finally, I'd like to thank you for your attention. Again, I would like to thank the organizers for this opportunity. I would welcome any comments and questions. Not sure. I'm still on. I think I can see the.
Thank you so much. That was really interesting. I have a question which may be based on a misunderstanding of thyroid as a so-called growth hormone. Does your work shed any light on, I mean, and given that, growth hormones are so prevalent in industrial farming and have, entered the environment, does this shed any light on environmental issues today?
Well, we haven't studied much. We did a little bit of work. There has been concerns. There's actually quite a lot of study on potential endocrine disruptors that alter thyroid hormone signaling. Although the current study is more focused on, well, most of the studies actually found chemicals actually inhibit thyroid. There's more inhibiting thyroid hormone signaling. They're less in terms of promoting thyroid hormone signaling. Now, growth hormone is a well-conserved thyroid hormone target in mammals. And thyroid hormone deficiency does cause severe short stature in humans. So I think it's probably related, although after birth, often actually, right now it's very hard to find a thyroid hormone deficient human baby or infants or child because of a thyroid hormone supplement.
Oh,
So
I wasn't talking about humans. I was talking about frogs. I mean, the great decrease in frog populations, for example.
Yes, there actually has some study that's related to inhibiting thyroid hormone. There has been proposal that perhaps endocrine disruptors may be contributing to the reduction in frog or amphibian level in general. Yeah, but we haven't done much. But one of the best known compound disruptor may be BPA and perchlorate.
Thank you.
Thank you.
Thank you for a really interesting talk. I have a question that you potentially answered, but maybe I didn't quite understand. And I wondered what was in the double knockout tadpoles, what was the actual mechanism of death?
Yeah, we do not know for sure, but the thing is, what we do know is adult organs are not are formed morphologically looks normal. However, functionally, whether they're fully normal or not, we don't know. So, like for example, the intestine, you can see the intestine structure resembles normal wild-type intestine, but the process of transformation is altered if we analyze carefully. The gills are still present, tails are still present. Whether they interfere with survival, we don't know. So we haven't studied. We think the resorption of larval organ may be important to ensure, if they don't resorb, they are not resorbed, they may interfere with proper organ function. Even some adult organs may have some larval tissues that maybe need to resorb. So we do not know yet, unfortunately. And honestly, we are not very good physiologists, so we haven't really looked into what cause of death. There have been suggestions we should look for the metabolism, look for food intake, but we just haven't done that yet. Very good question. Thank you.
Thank you.
I'll be in touch with you.
Thank you.